said, this has been going now for a number of years, and it's an attempt to sort of really bring out research into a wider community to understand what people are doing and to understand a little bit about the person themselves who does the research. So it is a great pleasure to have Jeff here today. Jeff and I go back quite some way. We probably shouldn't say how long, since that might reveal our age. And it's going to be great to just have this conversation, Jeff, a little bit about your past, how you got into Parkinson's disease, and then sort of concentrating at the end on the wonderful work you've been doing around co-pathologies, which isn't something we've really touched upon in this before. Most people think about Parkinson's disease and alpha-synuclein as one protein, one pathology. So it's useful, Jeff. So you're moved fairly recently.
Yes.
And so where are you based now?
I'm in Tempe, Arizona at ASU. Out of nowhere came a phone call from a headhunter 3 years ago, saying that there was this job I was recommended for. And I said, I was at Rush University Medical Center for 31 years, and
That's in
everything was
Chicago.
going swimmingly.
Yeah.
And,
So that's
really
in the
didn't
cold
have,
north, is it?
it isn't the cold Midwest. And but I said, I don't want to be a chairman. And she says, this isn't a chairmanship. I said, well, I don't want to take over a directorship and have to change the culture. She said, that's not this. So I said, what is this? It's to be the founding director of a neurodegenerative disease research center, which in my thinking prior to the phone call, that was the only job I'd ever be interested in.
Yeah.
And I thought, wow, that's nice, but you need to have resources if you're going to be building a center. And she goes, well, the Edson family have given $25 million endowment to your center. So I said, well, I'll be happy to check this out. And it was fantastic. I really underestimated the fantastic science that's going on in the Phoenix area.
Yeah.
And I'll just share with you one quick personal story, because I had gotten remarried about a year prior to this. And I said to my wife, I said, Janet, I said, I think I really want this job, but I'm not going without you. What do you think? And she goes to me, darling, when I fell in love with you, I thought I'd be stuck in Chicago the rest of my life. I want to go somewhere warm. So if you want to go to Phoenix, you can we'll go Phoenix together. And we are just having a fantastic time out here.
And so, of course, if anyone else wants to experience Phoenix and the wonderful place, May 2026 is when the WPC is
That's exactly right. Exactly right.
So, Jeff, perhaps we could go back to the beginning. So where was it you did your sort of undergraduate degree? What was that in? And then when did you go and do your next, your PhD or whatever?
So I got my bachelor's degree in psychology from Queens College of City University in New York. But
And that's
there
where you were born and grew up in New York.
I grew up in Forest Hills, Queens.
Yeah.
Right where they're playing the U.S. Open right now, about a mile from where they're playing the U.S. Open in tennis.
Okay.
And I was really not a very good student. I screwed around too much in my first 2 years of college. And my father, who was a chemist, said to me, you're more than welcome to continue down this path, but of course you're going to have to pay for the rest of your education. So what happened was I was in a developmental psychology class, and a professor said, I'm looking for volunteers to do research. And so I raised my hand. I figured, let me get some extra credit here. And we were doing silly science. In fact, I will tell you, Raj, and this you don't know about me in my career, but I have published the most seminal paper on the effects of olfactory bulbectomies, that's removing the structure that allows you to smell, on maternal behavior in male rats. It was just silly science. But let me tell you what happened.
Downhill since then, has it, Jeff?
But something dramatic happened, and that is right next to her lab was a new assistant professor moving in, and his name was Richard Bodner. He was doing pain perception and analgesia, and that I was interested in. Right. And he became my mentor. And I will tell, and I had a fantastic graduate career. So that was my senior undergraduate. And then when I got into the graduate program, and he was my mentor, I often think if his lab was on a different floor or a different building, or even on a different side of the floor on the same building, my entire career would have been different.
Yeah.
because I wouldn't have fell in love with science the way, because he was my mentor. It was just the most fantastic experience going forward.
So this, I
And
think this is a very interesting aspect that, I think a lot of people think their careers are plotted out in advance. And if there's young researchers here, they think I know exactly where I'm going. But it's often these sort of slightly chance encounters, which then really inspire
exactly.
you. And people who believe in you at a point in your career when perhaps there isn't that much to inspire.
Absolutely, absolutely. And I tell you, so I run a conference, as you know, every year. And last year, and I usually name each session after some hero of mine in our field. And I realized I didn't have someone named for the sessions that I have 4 students present.
Yeah.
We have all these international scientists, famous scientists coming up, but I have a session on the students. So I invited him, and I called it the Rich Bodner session on graduate education.
Yeah.
And he has graduated 35 PhDs, 35 masters, and is published with 250 undergraduates.
Wow.
That's how fantastic he is and was as a mentor.
So what did you do your PhD in your
So this is an important point, because my thesis was on vasopressin-induced analgesia. Vasopressin is a hypothalamic peptide that it usually controls water balance and urination. But we also found that it also influenced pain perception in experimental animals. And so the reason this is important is because I went to the Society for Neuroscience when I was the year prior to my graduating.
This is this huge meeting
with my PhD.
Where thousands of people go in neuroscience.
Exactly. As I like to say, it's like 30,000 of my favorite drinking buddies come, and we go to neuroscience meeting. And what happens is they have all these talks in different rooms, and outside the room is the name of the session that is ongoing at the moment or is about to start. So I'm walking with a buddy of mine, and I see brain cell transplantation.
Yeah. And
I go, that is the wildest thing I've ever heard. Let's go sit in here. Again, Roger, just a chance. If I wasn't walking around, if I was in poster sessions, this wouldn't happen. So I walk in, and there's a number of people talking. Anders Bjorklund, who's a hero of both of ours. And I think Barry Hoffer was also there. And there was a guy named Don Gash. And Don Gash was doing brain cell transplantations. He was taking the part of the brain of a fetus that makes vasopressin and putting it into a rat that genetically could not make vasopressin. And it started, and then the animal was able to stop drinking so much and stop urinating so much. He had normal water balance. And so I thought, well, what a match is that? He's doing vasopressin transplants. I'm doing vasopressin analgesia. And let's go and, let me see if I can get a postdoc there. So I go up after the session's over, I introduce myself. He happened to know my mentor. And I said, I would love to apply for a postdoc in your lab. And he goes, send me an email. I go back home, I send an email, brings it back out. I go give a couple talks, I'm meeting everybody. And he goes, I'd like to offer you a postdoc. I thought, fine. And I had 3 postdoctoral offers. I accepted his. And I went back to Queens. I defended my thesis, passed my thesis. Now I'm Dr. Jeffrey Kordower. And we packed up the car and drove up to Rochester, New York to start my postdoctoral fellowship. And so here's another.
And that was vasopressin tissue transplants, or did you move into Parkinson's at that stage?
Well, let me tell you how this happened. Yeah. So I walk into the office the first day, and I go, Don, I'm here. Let's go do vasopressin transplants. And he said a statement that changed the course of my life. Yeah. He said, we're now a Parkinson's lab, and you're the monkey guy.
Okay.
And again, if that didn't happen, I wouldn't be sitting here. It's again a total good fortune.
Yeah.
Now, good fortune is being prepared to accept good fortune. and
Yeah.
but our lab became one of the elite labs in doing brain cell transplantation in monkeys and in the Parkinson's models, and that's how I got started.
So that's how you got started in Parkinson's. And then sort of what was driving you early on was these sort of, because you've obviously very well known in the world of cell transplantation, which we can touch upon. You're very well known in the field of growth factors and trying to promote, repair of the dopamine system in Parkinson's. So was your ambition early on to really come up with better therapies? Was that really the sort of main drive of that early work? And during that period of time, what approaches did you take and what did you sort of learn, I guess, which will sort of set us up for where we're going nowadays with therapeutics?
So the reason I got into therapies is because I had a father who had god awful Crohn's disease and
Right.
pain and suffering, and he lived his entire life in pain. Really, it was just awful. So the idea of doing something for people
Yeah.
was a driving force. The other thing was, even though I was, a lousy student in organic chemistry, I was a whiz in the lab. I loved the lab aspect of things.
Yeah.
And being in a lab in graduate school replaced for me what I got in terms of playing sports. I mean, sports is a team effort. I play baseball,
Yeah.
and having teammates or other colleagues in the lab sort of replaced that need I had for that kind of human interaction.
Yeah.
And I started out, working with Don and monkeys, which I found to be fascinating. There were a million people doing rat work, a million people doing mouse work, but there were only a few of us doing monkey work. I mean, I do mouse and rat work as well, but
so this was right, and you've been very faithful to the non-human primate monkey models. So just whilst we're on that, I mean, what are the advantages and disadvantages of that? Because of course, it's very controversial around the world. Some people have very clear views it one way or another. Some people say this is much closer to man, so you should be doing these type of work. Other people would say, we shouldn't be doing this. So, I mean, obviously, you've been at the forefront of that. So people listening to this, what would be the advantages of using a primate model, and what sort of models did you have then, and what sort of models do we have now?
So I use both aged monkeys as a model of Parkinson's disease because the chemical that is depleted in Parkinson's disease, dopamine, in the nigrostriatal system, is significantly reduced just as a function of normal aging. Yeah. And Parkinson's patients is an age-related neurodegenerative disease, so there is that linkage there. And rodents don't show the changes in the system that aged monkeys do.
But monkeys don't get Parkinson's, is that right?
They don't get Parkinson's. I can tell you, in my career, there are times where I've had like 60 to 100 monkeys. And here in Arizona, I have 60. And at Rush, I had about 100. And I tested every aged monkey in their response to levodopa. And I found one out of all of these. And it turned out that when I ultimately looked at the brain of that animal, he had the most severe loss of nigrostriatal dopamine of any of the other cohorts. So I think it may have happened once in my career, but you're right. Okay. Parkinson's disease.
And why is that, Jeff? I mean, I have one patient who every time I see them in clinic, all he ever says to me is, why don't we just study the genetic differences between non-human primates and man, because man gets Parkinson's and humans get Parkinson's and they don't.
Well, part of the reason may be is that people live to be, 70, 80, 90 years old. Well, monkeys at 30 years old really have reached the end of their lifespan. Okay. It may be, but I will tell you, we may get to this in other parts of our discussion. So as a monkey ages, the levels of alpha-synuclein, which is a pathological feature of Parkinson's disease, and we'll talk about that in a little bit, increases as a function of age, but always in its normal monomeric form. When they say monomeric, in Parkinson's disease, the alpha-synuclein clumps. It's sort of like a garbage disposal stopped working, and you get clumps of this. But in normal aging, it is it's always the normal form, but too much of the normal form. So that may be there may be something that tips it over, and this monomeric form of alpha-synuclein becomes aggregated and more pathological and more toxic. And I think it's the machinery that normally clears bad proteins, what we call the lysosomal system or the autophagy system. I think that fails in Parkinson's disease. And while it's impaired in normal aging, it doesn't fail in normal aging. And I think that may be one of.
So if you could keep monkeys going for 50 years, you might see it. You think?
Yes, yeah. I think so.
So you were saying that the way in which you study Parkinson's, one would be using aging monkeys, and we might pick up on that because that's been very useful for some of the work you did on growth factors. And then the lesioning models as existed when you started were sort of chemical, I would guess, where you actually just put a toxin.
Well, we first started with a compound called MPTP. MPTP was discovered by Bill Langston and John Tetrut in California because a person came into the emergency room looking exactly like an end-stage Parkinson's patient. But the only issue was he was like 20 years old.
Yeah. And
it turns out that there were 7 of these cases around the California area, and it turned out that they were all drug addicts.
Yeah.
And so heroin's illegal, but if you change one bond on heroin anatomically, it is no longer heroin. So there's no law against it. So people were making these designer drugs, and it got contaminated with this compound called MPTP. And then we found that the greater we, not me, but the greater we found that if you give it to rats, nothing. If you give disease, not a great one, but if you give it to monkeys, you get full-blown Parkinson's disease. Okay. It's just, at least for the nigrostriatal degeneration. Okay. And so it turned out to be a fascinating model.
So that was your original model, was these neurotoxin models of Parkinson's.
Yes. Correct.
And that initial work with the toxins was really then used to inform work about cell therapies. Could we replace the dopamine system? And exactly. A little bit about growth factors, trying to regrow that system. Is that?
Yeah, that's absolutely true. So I was part of a team led by myself, Warren Alano, and Tom Freeman. Yeah. That was doing fetal cell transplants in patients with Parkinson's disease. Prior to that, we were doing work in monkeys with adrenal cell transplant. There was this insanity that occurred during our time, Roger, in which there was adrenal cells were shown not to work. And then there was this fellow in Mexico who said that it worked like it was the total cure of Parkinson's disease, and people went insane. No one was able to replicate that, but we tried that in non-human primates. Yeah. And what we found actually is that adrenal, and this sort of is a sidebar at this point of our careers, but it could induce the sprouting of residual host fibers in the brains of these monkeys.
And is that what led to the GDNF work, or was that sort of independent? So
that was independent. Marty Bone was the first to show that GDNF would. So she did adrenal cells in mouse, found the sprouting, and then there was some others that were using GDNF was found that it influenced dopamine neurons in a culture dish, and then people took it up into animals, and then we started doing the GDNF and the nurturing studies in non-human primates, and that led to a formation of a company called Serogene. We did 4 clinical trials, but it turns out.
And the non-human primate work, Jeff, I mean, how informative was that? So coming back to this original question, how useful are those type of experiments? So obviously the MPTP doesn't work in rats, so you can't do those type of experiments. So you can only use them in mice or monkeys. So for those listening, what would be the sort of things you can take away from the non-human primate studies that really help you or give you confidence to move a therapy into the clinic? And obviously these 4 clinical trials with GDNF and all the cell-based therapies, the trials that we've seen.
Well, there are a number of things that give you confidence. In fact, but you got to listen to the data. Because let me tell you where we didn't listen to the data. Let me tell you where we failed, and that's also informative. And that is, we use monkeys because the brains are bigger, and we know how much we need to put in of whatever we need to put in to get good coverage in the targets that we're interested in.
Yeah.
but there was also a voice in the company that kept on saying, well, it'll take too long and surgeons won't do this. And so we
But
started our
infusing the
initial
growth factors
trials.
into it. Yeah.
Right, yeah.
Yeah.
And we started our initial trials not putting in enough.
Yeah.
To give you an example, we put in our first trial 4 injections, 10 microliters each. It's like a droplet
Yeah.
on each side of the brain. Chris Bankovich is running a clinical trial now where he's putting in, instead of 10 microliters on each side, a milliliter each side.
Yeah.
And so that's a big change. So that's some of the things that are helpful.
So the value of the non-human primate was, so I mean, I'm not a great expert on this, but it's a monkey brain. How big is that relative to a human? About a fifth of the size? Would that be
about a fifth? About a fifth.
Which would be, and how
but
much
a mouse brain is like 100th.
Yeah. So what you get from the non-human primates is you get a scaling up. So you think, if I can get it to work in that size brain, there's more of a chance it'll work in a human brain. Whereas actually a tiny amount of these 10 microliters you're saying in a mouse or rat would have dramatic effects, but in a human brain,
that's right,
it's not covering anything. So it gives you confidence that you can cover the area. Do you think the system is more like that you find in the human, and does it enable you to study behaviors in a way which is difficult in
yeah so I was just going to bring that up. So the motor, you can have the animals trained on more complex motor tasks.
Yeah.
One of the big concerns, and this is something I'm changing my lab around right now, is I think many of us, and my lab included, are too nigrostriatal, too dopamine centric.
Yeah.
The major, we have things that can help people with the motor aspects of Parkinson's disease. Obviously, levodopa, deep brain stimulation, focused ultrasound now are all very helpful in this regard. But the things that get patients into nursing homes are falls and dementia. Yeah. And the ability to have high-level cognitive function testing in monkeys is something that we are working on very strongly right now to really address the non-motor aspects that are so debilitating and for which we don't have many treatments.
Okay. So those early days, you were sort of pioneering the monkey models of Parkinson's as you described, using toxins, and out of that work, you obviously led seminal studies on cell-based therapies for Parkinson's, and also really led the whole growth factor field around GDNF, and so that work sort of evolved. And I mean, one of the things which it might be worth just touching on is you've also been very critical, I would say, in reverse translation. So basically we do experiments in the lab and we take them to the clinic, but you've also been very much at the forefront of doing these trials, and then in people who've unfortunately succumbed to their illness or something else, look at the brains of people who died with Parkinson's or these experimental therapies, which I think is a great example of how you don't just sit in one silo, as Eli was saying at the beginning. You don't just sit in the lab, but you actually translate across, because I think some of that work has been highly informative. So I don't know how much that sort of post-mortem work you did informed what you did in the clinic, and that might lead us into this discussion on co-pathologies, I suspect.
Very, very, very much so. In fact, that is in many ways the most exciting aspect of my career, that no one has looked at more brains that have received growth factor therapy or cell replacement strategies than I have. And I publish every single one because I think these people were brave enough to allow us to operate on their brains. Yeah. Then I'm going to be responsible enough to publish every single one, even if it's, well, we've seen this again. Yeah. And although, we have seen, major differences between a person who died 18 months following transplant and one who died 10 years or greater following transplant, because the pathology of Parkinson's disease shows up in the graphs of those patients.
But those
that has changed.
Were very important, weren't they? So that first paper I remember you published a few years ago, we'll leave it at that, on the 18-month transplant.
Yes.
I mean, that showed the cells survived. So, I mean, for people who weren't in the field, this idea that if you just took fetal tissue from termination of pregnancies, put it in the brain of someone with Parkinson's, that it actually survived and made fibers and made some sort of connection. I mean, you were the first to demonstrate that, and that clearly showed.
I want to tell you that was one of the most exciting moments of my career. I'll take you 2 weeks prior to the patient, me showing that in the lab. But I come home and I see the answering machine blinking. And
Oh,
so I press the answering machine and I go, and I hear the neurosurgeon, Tom Freeman, my colleague, goes, Jeff, we just had a patient die. What do I do? So I call him up. I said, you do nothing.
yeah.
You take the brain, you put it in fixative, and you ship it to me. And then so we cut the brain, and then a very well-known transplant person named Tim Collier was in my department and just a couple doors down. And he goes, well, when are you going to stain the brain? And I go, I'm doing it this afternoon. He goes, I want to watch. And so we go into the lab and we put on, I took one section from each hemisphere and did it separately. I put it into a separate dish. And we did the last step. And then I just looking at it, and you could just see it happening. It's sort of like developing film, in which you can see the stain that you're interested in come from white into brown, and it was one of the most thrilling things ever. And so I go to take the tissue out of the dish and to throw it under a microscope, and Tim goes, "Let it go, 8 minutes," which is typically the time that we use. And I did that. Then we put it under the microscope, and there was the first evidence ever that a human fetal graft could survive in the brains of a Parkinson's patient. And it was just thrilling, just thrilling.
So that, of course, gives you great confidence that all the stuff you've done in the monkeys, which look very similar, you now could look at it in humans. And coming back to a point you made earlier was that, the limited lifespan of monkeys and obviously the limited time in which you follow transplants means that you don't get this long-term analysis, which you then subsequently went on to show in Parkinson's with transplant, showing that after 10 years or so, the transplants start to develop some of the pathology of Parkinson's.
Correct.
Which was unexpected, I would say, because of course you wouldn't really, because these are young dopamine cells, so why should they have acquired the pathology of Parkinson's? But I mean that was 2008, I think. So that
yes,
this post-mortem work has really not only confirmed what we've sort of found preclinically, but has opened up new avenues, new sort of findings, which has really galvanized the field.
Well, thank you. I don't know if you remember, Roger, you were chairing a session at a conference, and I think it was in Lund. And
I think it was in London.
At the last minute, you said to me, why don't you give a talk on what you've learned from your post-mortem cases? Yeah. So I just threw some slides together, and I got up and I said, I learned nothing because I've seen this all before in rodent and non-human primates. Yeah. And it was, I mean, I just got another one, maybe the last I'll ever get, that is 24 years post-transplantation. Yeah. And the patient has lots of surviving cells, has lots of Lewy bodies too, but lots of cells, innervation. Those grafts are going to live forever until the patient passes. Yeah. So that's a take-home message from cell replacement that I can make fairly strongly right now. The graft will not fail.
So these non-human primate works, which you've led on, they were very informative about cell-based therapies. They also informed about GDF growth factor approaches and some of the problems we had with delivery. But sort of moving towards this idea of co-pathologies, I mean, what I'm interested in is, when we started, alpha-synuclein wasn't even known as a protein associated with Parkinson's, and then it became associated with these Lewy bodies, which you talk about, the core pathology. And subsequently, it sort of gained a prominence. So obviously, alpha-synuclein defines Parkinson's pathologically, but obviously a lot of energy has gone into looking at treatments around alpha-synuclein. But what I'd be interested to know is coming back to some of the questions, the points you made about aging and alpha-synuclein. What do you think happens in Parkinson's? So do you think, so the simplistic view would be you get a bit older. Once you get beyond a certain age, alpha-synuclein changes in some format in some people. It starts to form toxic forms. It then aggregates. You get the first features of a prodromal Parkinson's, then you get Parkinson's, and then the disease progresses. And so if you can understand what that initial event is and you go after alpha-synuclein, in essence, you will be able to cure people. And that's the end of the story.
Right, except that's not the end of the story. Yeah. So let me just step back. So when we were doing our nurturing trials and we were thinking about...
So this is the GDNF growth factor.
Right, the GDNF-like molecule. Yeah. And we failed. And we thought, well, why did we fail? Could have been delivery as part, but it also, someone said, well, what is the putamen, which is the region that it liberates dopamine and is absent in Parkinson's disease, pretty much. What does it look like in a patient that had it was only 5 years post diagnosis of Parkinson's disease? Because that was our inclusion criteria.
Yeah,
you couldn't be in the trial unless you had Parkinson's for 5 years.
and that's
and we
overt
then
Parkinson's, motor Parkinson's.
Right,
Yeah.
exactly. And so then I looked back on what did the brain look like. Did a whole series of brains from 1 year post-diagnosis to 24 years post-diagnosis. And by 5 years post-diagnosis, almost all the dopamine was gone in the putamen.
Yeah.
So we had to figure out a way to get earlier than 5 years. So I started to look at patients from the Rush Center on Healthy Aging that were nuns and priests cloistered in nursing homes that got clinically tested every year, and then Alzheimer's brain donation programs. And I found that those patients that had motor impairment but not sufficient to diagnose Parkinson's disease, that they had diminished dopamine, diminished dopaminergic cells, diminished dopaminergic innervation, and they also had a very strong synucleinopathy. The synuclein in those patients was very high. In fact, more than PD cases, because the PD cases had cell loss, and these patients hadn't had cell loss yet. And so
did you think they hadn't had cell loss, or they were just resistant to the pathology?
I think they hadn't had cell loss yet. Okay. Well, I mean, they had cell loss. Let me step back. But it was intermediate. It wasn't at the same level that you see in Parkinson's disease.
Okay.
So they had cell loss, and they also had decreased innervation. And so, but to get into that study, the patients had to have a clinical no motor impairment in Parkinson's disease. That was a criteria. And they had to have synuclein. Yeah. So, because that's what, after all, isn't that what synuclein's all about?
Yeah.
So it always bothered me that I made that choice in including those cases. So when I moved to Arizona and was setting up the lab, I want people to be collecting data. I said to one of my faculty, Yaping Chu, I said, I want you to read just on the clinical. So the only thing you need to do to get into this trial was to have the clinical. And it turns out we were able to have two different groups. Yeah. We had clinically mild motor impairment with synuclein, mild motor impairment without synuclein. And it turns out that they're absolutely identical in almost every single measure that we did. Cell loss, innervation, exoplasmic transport, everything we looked at, there was no difference between the mild motor impairment with or without synuclein.
Okay.
So then
what you're saying is that as you get older and you get mild motor impairment, previously you had said that's due to an alpha-synucleinopathy because actually you only selected on alpha-synucleinopathy. Whereas in fact, if you take all comers and say, I don't really care, just anyone with mild motor impairment, some of those people could have that in the absence of an alpha-synuclein pathology.
Absolutely.
And what was that pathology then?
The pathology was a protein called tau, which is what we think of in terms of Alzheimer's disease.
Okay.
But there was a tauopathy in every single one of the mild motor impairment groups, whether they had synuclein or not. Okay. And also in 22 out of 24 of our cases that had Parkinson's disease, all had this protein tau. And this protein tau is involved in a lot of different activities, mainly keeping the axon intact in certain neuronal systems, especially long projection neurons. Okay. And so we believe that these co-pathologies of synuclein, tau, and amyloid together are what causes the ultimate deficits that are seen in patients with Parkinson's disease, especially those that are cognitively impaired.
Yeah. So if we go back, so you know that study which you just reported, so tau is common to all of it. So you could say Parkinson's disease is a tauopathy then, is it? Because that seems to be the primary pathology in more cases than the synuclein.
Crazy when I say that. So especially this
So if I just say, let's flip it around the other way and say, let's take, I don't know, 40 people or a group of people with well-established Parkinson's disease who have then died of their Parkinson's. So let's take the post-mortem from the transplant cases. So do most people who have had Parkinson's for many years and then die either from Parkinson's or something else, do they have tau pathology in their brain as well? Or is it that tau pathology is early, seems to trigger something, synuclein comes along, that dominates, and then that's
yeah, we really don't know very well. It's hard, first of all, to get young Parkinson's cases because they survive, and those materials are very difficult to get. I know I had this very discussion with a colleague of ours, Matt Farrer, very recently, and he believes, so if you put LRRK2 into a mouse, so you make a transgenic mouse,
which is one of the key genes involved with Parkinson's.
Exactly, it makes tau.
Yeah,
it's a tauopathy before it's a synucleinopathy.
Okay.
And so there are a number of these experimental models that suggest that tau could come early and then work together in to cause a degenerative process. So the fact that we have multiple pathologies now, of course, the way I flip my brain is, so what do I do therapeutically? If I got three,
get to that, Jeff. So if I flip it round and say, so there are people who have tauopathies, so that these are people who have sort of another type of condition, frontotemporal dementias and progressive supranuclear palsy. Some of these, have Parkinsonian features. Do they have an alpha-synuclein pathology as well, or do they just have a pure tau pathology?
They tend to have more of a pure tauopathy.
Okay. So Parkinson's is unique in having this tau going to, or tau and synuclein.
Right.
Okay.
So
and do you think there are other proteins? Because you mentioned amyloid. So obviously as we get a bit older, and I would think most people by the time they're 80 or 90 will have a bit of Alzheimer's pathology anyway. So do you think in Parkinson's, we talked about co-pathology, so we have synuclein and tau, we'll come on to the therapeutic implication. Do you think it's other proteins? Well, do you think there's amyloid, which is so heavily associated with Alzheimer's, and at the moment is getting a lot of publicity because of the antibodies in the Alzheimer trial showing some sort of signal of efficacy? Do you think amyloid is also involved?
I do think amyloid is involved in certain cases. No, we talk about Parkinson's disease when we should be talking about Parkinson's diseases, because they're all a little bit different. But I do think that in certain individuals, yes, amyloid is a problem.
Because there has been this slight move recently, as you all know, to get rid of the name Parkinson's.
Right.
And to start talking about neuronal alpha-synucleinopathies. So, but your argument would be that would be a retrograde step, because it implies it's all in one system or in one cell with one protein, and that's not the case.
And it's not only just neuronal. They call it neuronal synuclein disease. There's some glial changes as well. So there's a paper that just got accepted this week that I'm a co-author on from the leadership of the Movement Disorder Society. And we make that very case that we don't think that neuronal synuclein disease is the right way to go.
Okay. So we've now arrived at this point where we think Parkinson's it's clearly age-related. It's clearly related to synuclein. It's got a component of tau, and there may be other proteins in it. So, and all of that has been informed by your post-mortem work. And then if you want to model that in animals, so one of the questions we actually received was, which isn't directly related to this, but, when you've been developing treatments, which is where we will go with this conversation, it says, all of the treatments have failed. So your preclinical models weren't useful because they didn't predict that they would, they should succeed.
No, because we've never modeled the co-pathologies. Right. So I have a paper we're just beginning to write where we put alpha-synuclein preformed fibrils into the striatum of aged monkeys that have amyloid. And then I have this fantastic model of tauopathies in monkeys. It's called AAV double mutant tau. Okay. And I put initially for our Alzheimer's studies, we put this into the entorhinal cortex, and
And
it
this you
spreads all.
directly inject. So this is a virally delivered protein which you inject into the brain.
Correct.
Yeah.
And so for the Alzheimer's studies, it spreads all over the connectome, the entorhinal cortex connectome. It goes to hippocampus, goes to retrosplenial cortex, it goes to occipital cortex, and it's just amazing.
This so you inject it into one site in the monkey brain, and it just spreads down all the connections of that.
And it forms tangles. It forms genuine tangles, and it evolves over time. We have a time course of this that causes cell death. And so what we're now doing is taking that virus. So we put the preformed fibrils in the striatum. We take that virus and I put it into Nigra.
Yeah,
causes tremendous cell loss.
Okay.
And in fact, if you put them together, the alpha-synuclein and the tau, you get more nigrostriatal degeneration than either one alone.
Okay. So you think the models may have failed because whilst they were a good approximation of Parkinson's, they were simplistic. So obviously the toxin models were just saying, if I kill dopamine, can I replace it?
Correct.
And that's what the cell therapy is. And obviously we've discussed that in the past in various other spotlights, and we've discussed it here that you can get some success. Growth factors are harder, but you do partial lesions, old monkeys. But again, it's not really Parkinson's. And, you know, some of the failure may be because of volume delivery, as you say. Some of it may be because the models have just not been good enough. And actually, all of our preclinical testing of drugs is normally done either in cells or in
rodents.
Or rodents which have alpha-synuclein preformed fibrils as the model. And so you're saying, I think we need to be a bit smarter and actually do dual, at least tau and synuclein, to better mimic what we see in Parkinson's, and then go back and revisit these therapies.
Exactly. But the question is, what therapy? So you have all these antibody studies that have tried. First of all, the pharmacokinetics of these drugs are awful, just awful. So you get very little into the brain, and you're just trying to reduce synuclein. Or in other studies in the Alzheimer's space, they just trying to reduce tau, but they're not trying to reduce both.
Yeah.
So how can we do that? Yeah. And we have a fascinating new program that we're just starting, and it's a trophic factor called irisin. This is discovered by Bruce Spiegelman at Harvard, who's a brilliant biochemist and collaborating with us now. Irisin, you know, the one thing that can slow disease progression is exercise. Yeah. And so when you exercise, irisin is released from muscle, crosses the blood-brain barrier.
Yeah.
And Bruce and in collaboration with other colleagues and with me has demonstrated that irisin will clear synuclein, will cause in synuclein models, will prevent the every motor test that Ted Dawson did in this one study was reversed with this drug. I've demonstrated with Bruce that tau is reduced and completely eliminated in cell culture. We're doing rodent studies now and then we're doing it in age. And he's also shown that amyloid is reduced with this irisin. And the key point here is you inject it once intravenously and it lodges in the liver, the virus. Right. And it doesn't do any liver damage. There's no damage to the liver, but it sits there and it secretes this protein. So it's a one-time treatment that can affect all three of the major misfolded proteins. And that is what we're really excited about.
So that is obviously a new therapeutic, and part of its advantage is that it actually targets more than one protein species. So it doesn't just go after synuclein, it goes after all of them.
Yes, exactly.
And so, so thinking forward, should we be doing more non-human primate modeling in the way that you say, and then testing combination therapies? So obviously the field in neurodegeneration has slightly moved into a sort of one protein does things. So in Alzheimer's disease, I've always said, Alzheimer's disease has amyloid and it has tau, but all of the antibody therapies currently being trialed are the ones against amyloid. And, why aren't they combined with tau, given that's also the pathology? So your view would be that we should be doing modeling using two proteins, and then we should be using combined therapeutics or finding new agents, as you have, which can target multiple protein species.
I definitely think we should do that. I can tell you it is somewhat complicated because if you're doing combinatorial therapies, the FDA is going to make you do safety and tolerability of them individually and together. So this enhances the cost and the time and all of that for that. But you know what's more expensive than a failed clinical trial is more expensive than doing the preclinical work.
Yeah.
And so I think that is definitely a way to go.
So basically, we should better model the disease using non-human primates and multiple proteins. We should be combination therapies or finding new therapies that don't just target one protein.
Yes.
So some of the questions which have appeared relate to blood tests and identifying POCs ahead of, overt symptoms. And obviously, there's been a lot of interest recently in this seed assay with alpha-synuclein.
Mm-hmm.
So do you think that would also have impacts on how we think about it? Do you think we should be thinking about whether we should be developing an assay which looks at multiple proteins in the population as they get a bit older? So we should be screening the whole population around these proteins so that we can start to see who's at risk and then use that for your new therapeutics.
One of the things that's being promoted out here in the Valley of the Sun is by a person by the name of Eric Ryman. He saw the king of Alzheimer's disease out here. And he's very interested in what we call prevention trials.
Yeah.
So what he's promoting is that, everyone, when they turn 50, they go see their doctor and they take a blood, which will then, right now, the seeding assay is a CSF assay. But soon we'll have blood assays. I'm totally convinced of that. And so if it comes up that you likely will have Parkinson's disease on this blood assay, then you go and you get PET scans or DAT scans. So you get a DAT scan, which measures dopamine. And if that's reduced and you have the blood marker, then you can start treatment even if you have no symptoms.
Yeah.
Especially for something like what I'm proposing with a gene delivery of virusin, because you only got to deliver it once.
Yeah.
And so that is the prevention trial model that is being proposed at this time.
Okay. And this irisin gene therapy, that's not something you need to have the gene therapy. If I went crazy in the gym, which is very unlikely, I couldn't produce enough irisin myself to overcome the problems which the gene delivery produces, concentrations which are way in excess of that you can do yourself.
Well, but people who are just just recently diagnosed, who are actively exercising, they have slower progression than subjects who don't exercise. So you can get some benefit just from the exercise itself.
Okay. And so one of the questions has been around younger onset disease. So obviously quite a few people on this call and quite a few people who will listen to this will say, average age to get Parkinson's is 70, but I'm 40, I'm 50, I've got it. So do you think that so obviously there is this, unknown, if you like, about age. What is aging? What does aging do to the brain, which makes you more likely to get this condition? And do you think if we do this approach, we screen the population over the age of 50 or 60, we do this sampling, we find who's at risk, we do a DAT scan and such like, do you think those treatments which will be the same for people who have younger onset disease? Obviously there are the rare Parkin patients where there are, people are looking at Parkinson's therapy.
Yeah.
But do you think younger onset Parkinson's will be suitable for these therapies as much as the older, or do you think
I do. I do. I will tell you that I haven't had access to brains to study from young onset Parkinson's cases. They're very rare and difficult to get.
Yeah.
So I can't definitively say anything, but there's no reason that I can think of. And so the other thing is you can take the young onset patients and you could do amyloid and tau PET scans.
Yeah.
And you can say, well, did they have these other, misfolded protein pathologies? And if the answer is yes on PET scans, then you could treat them. And there's no reason to think that a young onset patient should respond differently than a run-of-the-mill idiopathic.
Because just as it happened, I was talking to someone at one of our research institutes here, and they were saying that they've done this very clever cryo-EM, this incredibly high caliber analysis of Lewy bodies, and they're exactly the same in Parkinson's dementia, and people with dementia with Lewy bodies. So it implies that whatever ultimately happens in all of those diseases is the same. And so it's always been this question of whether there are separate diseases or whether they're all the same disease. They just have different kinetics, so they're driven at slightly different speeds. So young onset patients may have something more abnormal in the alpha-synuclein pathway, or tau pathway, which is driving it. But ultimately, if that's true, then all of the therapies should work for all patients, regardless of what they've got. And that would be very much your view.
That would be my view. That would be my view.
So just in these last few minutes, Jeff, I mean, it's been a fascinating, discussion of how you got into Parkinson's disease, the work you did on cell and genes, but the fact you never stay still, just don't concentrate on one thing, you move forward and you take the work forward. So, I mean, there are still questions appearing about, the value of the models. So if I was to play the devil's advocate, I would say, well, we've got alpha-synuclein antibody therapies, we've got amyloid antibody therapies, we've got tau antibody therapies that are in clinical trials. So do I really need to do any more animal studies, or why don't I just get a brave group of people and chuck all of these antibodies at them and say, let's just go for bust with that? So do you think that preclinical testing is necessary? And I suppose just thinking back to your work on irisin, I think this is a very good example of where do you think that the primate studies have taken us with that? How have they really helped us?
Well, we're just getting started on the primate studies with irisin. So we haven't done that yet, but we just wrote a grant and we're finalists for the Sparks NS program to study irisin in the non-human primate models. I think it's still extremely valuable for a number of different reasons. And one is that things are worse when you have co-pathologies. And if you have something to clear them, there's a greater chance that it's going to work in patients. The other thing is, I'll tell you what's interesting. As you know, Roger, I'm a founder of a company called Kenai Therapeutics that is about to get their IND approved for doing patients with stem cells for Parkinson's disease. And, when we put in the IND, they requested no monkey studies. The only thing they requested in terms of we have to do additional studies. So we did safety, tolerability, and, all the manufacturing stuff. But the difference, all they wanted to know is if our device that we're going to deliver the cells will deliver the cells the way we think they will. And so we did a small monkey study and looking at 7 days and 32 days of our cells, and it did just fine. So it's interesting that the
regulators,
the FDA is changing. It's changing. You used to have to do 2 species of safety and tolerability, one being of a large animal. They don't request that anymore.
But I mean, just to sort of conclude, I mean, I think you've made a very strong argument as to why some of these experiments are still needed. I think one can move away from them, and I think perhaps before our time they were done a bit liberally, non-human primate studies, when they could have been done in other species. But from they do provide valuable information not only about scale but about efficacy. And I think your point is well taken, that a lot of the reasons why our clinical trials fail, where our preclinical work, is that the model we have is not the model of the clinical disease we're looking at.
That's right. And I will tell you, one of the things that I insist on with all my students is that they go see patients. Now, for you on the Zoom, I'm a PhD, not an MD, but I know a lot about Parkinson's disease because I go see patients. And when I study atypical Parkinson's disease, such as multiple system atrophy, I went and I saw patients. And because you need to understand what the clinical entity is to best model Parkinson's disease and other diseases going forward.
Well, we're sort of almost out of time. I just want to thank you, Jeff, for for all your work over the years, which has been an amazing collection of sort of seminal studies. And one of the things I've always sad about is people don't spend enough time on history and how we arrived at where we arrived at, and some of the seminal, so your post-mortem work, all of the GDNF in aged monkeys, the nurturing, and then learning from that and going forward. And I think that's a very important message, this iterative process. Just because the trial doesn't give you the result you wanted doesn't mean it's a failed trial. It means you should learn from it. And you've been absolutely at the forefront of taking those findings and thinking, well, why haven't they worked? Perhaps the therapy doesn't work, but what does that mean? And I think, we've now ended up in a position where you can say, well, actually, in retrospect, everything I've been studying has been very helpful, but I've used the wrong models and I've got better models. And I think
Right.
it's very instructive also to hear about the complex pathology of Parkinson's disease, because there has been a slightly sort of reductionist approach that, as I've been saying, that everything comes down to one protein, one
Right.
conformation of it,
Right.
and that's the end of it. Whereas I think, the complexes of the human brain and aging and these multiple pathologies is really where it's at. So I want to thank you so much for for everything that you've contributed. Thank you very much for this conversation.
Always great fun, Roger.
Yeah, so I've really enjoyed it. I just want to thank again our sponsors, Supernus Pharmaceuticals and Mitsubishi Tanabe Pharma America, for the support for this, and to encourage everyone to join us for the next one of these, which will be on Tuesday, October the 8th. And that will be at the same time, and the speaker then will be on a very different topic. It will be Michelle Hugh from Oxford, who will be talking about prodromal Parkinson's, which, based on what we've had tonight, this afternoon, this morning, wherever you are, this perhaps should be the group that should be getting Jeff's new therapy, as well as some of the therapies we've already discussed. So thank you very much for joining us today, and we very much hope you enjoy this and look forward to seeing you again in October, and failing that, seeing you in Phoenix in May of 2026. So thank you very much.